Hello guys, welcome to BioEd. I am Aman from BioEd and welcoming you to a new video tutorial on cells of the immune system. This is going to be the part 4 of the cells of the immune system. In the first 3 parts we have discussed about phagocytes and mononuclear phagocytes. In the second part we have discussed about granulocytes, and in the third part we have discussed about antigen presenting cells. This is going to be the part 4 of the cells of the immune system, in which we are going to discuss about lymphocytes. Their different classification, their job, their role, everything we are going to discuss in this video. So before we get this video started, do make sure you hit the subscribe button and turn on notifications that you don't miss any videos in the coming future. So let's get the video started. Guys, now let us discuss about lymphocytes. So what does the word lymphocyte mean? The word lymphocyte is made up of 2 words. First is lympho and the next is cytes. Lympho means lymphatic system and cyte means cells. So the cells of the lymphatic systems are known as lymphocytes. They are also present in the blood but mainly found in the lymph nodes. So these are non-granulocyte cells, that is, they don't contain granules in them. You can see a small basic diagram of a lymphocyte over here, that they have a really large nucleus and a small ring of cytoplasm surrounding the nucleus. The cytoplasm is really less in the cell. Okay, the next is they are the members of the WBC. They are the cells of the WBC, they are the members of the WBC. And they are differentiated, they are divided into 3 different parts. The first is B cells, the second is T cells, and the third is natural killer cells. So now let us discuss about all these 3 different parts in detail. So first we will be discussing about B cells. So now let us talk about B cells. These B cells are the cells of the adaptive immune system, that is, they function in the adaptive immunity. So these B cells, their main function is the recognition of antigen, that is, antigen is a thing that can trigger an immune response. Their main function is the recognition of that antigens, that is, they recognize whether any antigen has entered in our body or not. Next thing is their origin. They are originated in the bone marrow. They are originated in the bone marrow, and then from there bone marrow they travel to the spleen and other lymph nodes, and basically these B cells are concentrated in the lymph node. So now you can see a basic diagram of a B cell over here, in which you can see a big nucleus, and on its surface of the B cells they contains B cell receptors. On these B cell receptors, what they do is they they recognize the antigens by the B cell receptors. So this is the antigen, this comes on the B cell receptors and it gets recognized by the B cell and it gets internalized, that is, it gets inside the cell, and this antigen is processed into various small parts by the help of receptor mediated endocytosis. This is the process by which the antigen enters the B cell and gets processed. By the help of endocytosis, this antigen enters, and then this antigen is presented on the surface by the help of MHC2, that is, major histocompatibility 2 class 2 complex. This is the MHC2 compatibility complex. On its surface this antigen is again presented. Why it is presented on MHC2? It is presented on MHC2 so that CD4 or T helper cells can come and recognize. Suppose this is a T lymphocyte helper cell. It comes and it can recognize this antigen on the surface and thus form an interaction. As soon as it forms an interaction, as soon as it recognizes the antigen by the MHC class 2, it starts the secretion, the B cell starts the secretion of lymphokines. The B cell starts the secretion of lymphokines. These lymphokines are chemical molecules that help in the process of mitosis. Why mitosis? Because they start the signaling of increasing the number of B cells in the near area. They start the process of, first is they start the process of mitosis, and the next is they start conversion of B cell to plasma cell. They start the conversion of B cell to plasma cell. The plasma cell, what the plasma cell does is they start the production of antibody. They start the production of antibody. So why does it start the production of antibody is the antibody is used to counter the antigen and to recognize, and the antibody that is produced over here is specific to the antigen that at the starting the B cell has recognized. So this was all overview about the B cell. Now we will be discussing about T cells. So now let us talk about T cells. So these T cells are involved in the production of cellular immune response. That is, they help in the cellular immune response. Whereas if you see the B cells, they were involved in the production of humoral immune response. Why humoral immune response? Humoral means blood immune response. So why in the humoral response? Because they secreted the antibodies, and these antibodies were secreted in the blood by the plasma cell. That is the conversion of B cells to plasma cells when lymphokines were secreted. Okay, so why the name T? Why are they named T cells? Because their site of maturation is the thymus. Thymus is the site of maturation. That's why they are known as T cells. Okay, so what is their main function, main role? So the main function of these T cells is that they recognize the antigens that are bound only to MHC complexes. They recognize the antigens that are MHC bound. That is, only they recognize the antigens that are processed and presented by the major histocompatibility complex. So these T lymphocytes are of three different types. The first type are helper T cells. These helper T cells, they recognize the antigens that are bound to MHC2 complex. They recognize the antigens that are bound to MHC2 complex. We have studied earlier in the case of B cells that they have the MHC2 complex, and on their surface these helper T cells come and recognize. They are also known as CD4 cells. The CD stands for cluster of differentiation. C for cluster, D for differentiation. It is a type of ID that is given to cells to recognize. It is a type of phenotypic character that the cell has. The next are cytotoxic T cells. These are the cells that are activated and proliferated. These T cells, when get activated and proliferated, that get converted into cytotoxic T cells. These are very helpful in the monitoring and controlling of the viral infected cells. So let me give you an example. Suppose this is a cell which has been infected by virus. So this virus, what it does, gets into the nucleus and starts producing viral proteins. So on these cells, a normal cell, they have MHC class 1 complex. So on the MHC1 class complex, what this cell does, this cell starts producing viral peptide, viral protein, and these viral proteins are shown on their surface. So it must be seeming a bit complex. So basically what it is that this normal cell gets infected by a virus, and the virus starts producing proteins, and this protein is presented on the surface by MHC1 complex. So what it does is this cytotoxic T cell comes over here and recognizes on the MHC1 complex that this antigen is being shown, and upon interaction it starts secreting cytotoxins, that is the toxins for the cell, and these cytotoxins start a series of events which follows apoptosis. What does apoptosis mean? Apoptosis means programmed cell death. That is, this when T helper cell comes and interacts, it starts the process of apoptosis and the cell dies. That's how the cell removes the elimination of virus infection or anything. The next are memory T cells. As the name suggests, memory T cells, these cells contains memory in themselves, that whenever any next time if the infection comes, these memory T cells are there and they recognize the infection and easily they remove the infection. So this was all about T cells. Now we will discuss about natural killer cells. Now we will be discussing about natural killer cells. As the name suggests, natural killer cells, the word killer in these cells tells about these cells that they have the killer activity, that is, they just go and kill the cells. So they mediate the innate immune responses, that is, they are not a part of the adaptive immune response, but they are a part of the innate immune response. They are closely related to B and T cells. They resemble closely to the B and T cells, and they do not express antigen specific receptors on their surface. They don't have antigen specific receptors on their surface. Okay, they are really efficient cell killers, that is, you can see the diagram here. These natural killer cells on their surface have they express inhibitory receptors. On their surface, these black receptors you can see, these are the inhibitory receptors that they have there on their surface. So whenever a virus infected cell or any cell that is injured, it comes and it forms an interaction by their MHC1 complex. They have the MHC1 complex on their surface, and as soon as they form the interaction, these both, what it does, the natural killer cells, suppose this cell and this cell, suppose it gets an interaction. So what these natural killer cells does, it starts the process of killing, and it destroys the cell, and thus tries to remove the pathogen or virus infected cell or any other cell that isn't working properly. For example, a tumor infected cell or a virus infected cell. So that's how it removes the process, that's how it removes the infection of our body. These are also known as CD16 cells. They have the cluster of differentiation 16 on their surface, whereas the T cells had cluster of differentiation 8. Okay, so this was all about natural killer cells, and this was all about the lymphocytes. So in this video we discussed about the lymphocytes and their various 3 different classifications and all various different things. So I hope you guys liked the video, so please drop a like on this video and also comment, and also make sure you subscribe to the channel and also turn on the notifications. So meet you in the next video. Bye bye.